Hello everybody, I'm Dr. Amit Sarkar, Assistant Professor Biotechnology at the Nehru University of West Bengal, and this lesson is being prepared under third FIP organized by UGC-HRDC DAVV Indore, and I'm very much thankful to Professor Namrata Sharma, Director UGC-HRDC DAVV Indore, and the entire coordinator team. So today in this lesson, we'll be learning about the basic functional differences between B and T lymphocytes and also their interactions for effective immune response. In the last class, we have seen that the B and T, both the cells are originating from the bone marrow cells to the process of hematopoiesis, but the B cell also mature at the bone marrow cell, whereas T cell mature at the thymus. Now, B cell, after leaving the bone marrow cell as a mature B cell, they're having a membrane-bound antigen receptor for antigen molecules, which is a glycoprotein in chemical nature, and which is known as antibody, membrane-bound antibody, whereas we have membrane-bound T cell receptor for the T cells. And let's see what they do with these antibody molecules or the receptors. So if we focus on the B lymphocyte at first, the naive B cell, that means the B cells which are yet to encounter any antigenic challenge, they for the first time after binding to an antigen molecule, specifically with this membrane-bound antibody, they will be dividing rapidly and differentiates into two kinds of cells known as plasma B cells, also effector cells, and memory B cells. So naive B cells binding to the antigen molecule, dividing and differentiating into two different kinds of cell, plasma B cell and memory B cell. If you want to see the differences between in these two types of cell, plasma B cell, which is also known as effector cells, effector B cells, they have a very short lifespan, but they produce secretory antibody. They release secretory antibody, and these secretory antibody molecules with the same specificity, they will bind to this antigen, to these kind of antigen molecules wherever they are found in the system. They will be mixed with blood and lymph, and they will reach every corner and will try to bind to these type of antigen molecules. Okay. On the other hand, memory B cells, they do not have any capability to release any soluble antibody molecules, but they will be remaining there in a large number with the membrane-bound antibody, the same membrane-bound antibody as their parent B cell in a large number. And their lifespan is great. They will be there for a long time, and they're kind of responsible for our lifelong immunity and also basis of principle for the vaccine development as well. So now T cell receptor has a kind of a peculiarity. T cell receptor cannot recognize antigen directly or free antigen just like B cell. They only can recognize and bind to an antigen only when it will be complexed with a protein known as major histocompatibility complex or MHC, and the whole process is known as antigen presentation. So who is presenting here? This B cell, after binding to the antigen, that internalizing it, processing it, and presenting a small portion of that microorganism, a small peptide complexed with the MHC molecule. Okay, so they are antigen presenting cell or APC. There are other antigen presenting cells such as macrophage and dendritic cells. So T cell now can recognize, bind to this complex MHC plus antigen and can recognize this foreign antigen. After getting activated, after getting recognized and activation, these T cells will release chemicals which is known as cytokine molecules. These cytokine molecules in turn will bind to the B cells. So now B cell will be activated. So the entire scheme, if we take into account, the naive B cell with this membrane-bound antibody for the first time binding to an antigen with specifically, and they are internalizing the antigen, processing it, binding it to MHC molecule, and this complex MHC plus antigen is being presented to the T cell. I will talk about this helper T cell MHC2 classification later point of time. So now the T cell with this T cell receptor molecule is recognizing and getting activated and releasing chemicals known as cytokines. And these cytokines in turn is binding to the B cell molecules, B cells, and the B cells will now get the signal to divide rapidly and to differentiate two kinds of cells known as memory B cell and plasma B cell. And just we have seen before that this plasma B cell will be releasing antibody molecules and will bind to the antigen and try to neutralize them. On the other hand, these memory B cells will be there for providing us the lifelong immunity for any particular microorganism. So that's all about this particular small lesson, and I'll be posting the link in the chat box for a quiz, MCQs, and the home assignment. And also, I will be posting a link for the Google Meet where we will be able to discuss these things and we'll be able to address if you have any questions in this regard. So thank you very much for listening. Thank you.